Simon Conway Morris has studied the fossils for over 30 years.
It's almost as if you've gone to another planet, you've been given a fishing boat and a net, and you've been allowed to throw that net over into the deep ocean, and you had no idea what was going to come up.
Some of the Burgess Shale creatures were familiar.
And here we've got one of the trilobites. We see the delicate soft parts also preserved.
Trilobites are extinct arthropods, creatures with external skeletons. Today's arthropods, like crabs, lobsters, insects, and spiders, are all descendants of creatures like these. Other Burgess Shale animals were bizarre, alien-seeming. An animal with 5 eyes and a long, retractable nozzle. One with long, sharp spines protruding from its back. Another with a circle of prongs around its mouth. And yet, as alien as these creatures seem, they are also surprisingly familiar. Like living animals, they have bodies with heads, tails, appendages. Specialized segments performing specialized functions. All the basic body plans found in nature today are here. Every animal that has lived for the last 500 million years has come from tinkering with these initial designs. We might even see our own ancestor here.
And maybe this is the crown of the Burgess Shale. This is Pikaia.
A tiny creature, Pikaia is one of the rarest fossils from the Burgess Shale. It's the only one with an internal nerve cord resembling a spine. That might mean that creatures like Pikaia were the earliest ancestors of all animals with skeletons.
The idea is that this might be the precursor of the fish, and so ultimately, through a long evolutionary story, ourselves. Why does a Cambrian explosion matter? It matters for lots of reasons. And basically, it's part of our history. It's where we came from, and that matters very much. This is the time when the animals first appear. We look back and we can see part of our history unfolding.
So what do we learn by looking at 600 million years of animal history? Evolution's tinkering with mammalness to make whales. In the same way, it's tinkering with fishiness to make tetrapods. And it's tinkering with animalness to make all the different body plans that we see. All these different creatures are variations of the same theme, restated over and over again. The question was, what was evolution tinkering with? One of the remarkable discoveries of the last 20 years is that evolution's not tinkering with the bodies. It's tinkering with the recipe, the machinery that builds bodies. What is that recipe? What is that machinery? It's the genes.
Fossils record the changes in animals' bodies over time. But just how bodies change was unknown. The search for the genetic mechanism of evolution took most of this century. When scientists finally found it, they were astonished by just how simple it was. One of the key players was Mike Levine.
I was, I guess, kind of a weird kid. I always liked bugs. We had a nice big backyard, and I could go back there. It was kind of a sanctuary, and I played with bugs, dissected them, manipulated them. That's really the most pleasant memory I have.
Levine's affinity for bugs led to his study of biology. One insect in particular became an object of fascination.
They have a quick generation time, and they have lots of pattern. I mean, you wouldn't know it if you look at a distance, but when you look under a microscope at an adult fruit fly, you'd be astounded by the number of bristles, the intricacies of their wings, the patterns of their eyes. But the embryos are something else. I do love the embryos.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One of the first people to study these questions was a 19th century naturalist named William Bateson. Bateson wrote that animal skeletons revealed an underlying structure of repeating segments. He also observed that animals occasionally developed with some segments in the wrong places.
Insects with legs in the wrong place, crabs where a claw was transformed into a leg, pythons with extra ribs, or frogs with extra cervical vertebrae, and all these sorts of things.
To Bateson, these developmental errors meant that the underlying blueprint for the animal was being disrupted. He had no idea how it happened, but he suspected that these random changes might provide the fuel for evolution. By the 1940s, scientists working with fruit flies had learned how to cause disruptions in the developmental blueprint by dousing growing embryos with radiation and poison.
And so when they did that, they found flies with changed wing structures, changed legs, and these very special flies that have one part of the body in the wrong place, or a copy of a normal part of the body in another place.
The scientists had triggered the changes by damaging the fly's DNA. Within each cell of the developing embryo is a chain-like molecule called DNA. The experiment showed that DNA was somehow causing the embryo to divide into segments. But how? Scientists were just beginning to grasp that the DNA itself was made up of segments, called genes. The question was, how did the genes shape the body? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body.
And as you looked at these genes, he said, this one affects this part of the body, this affects the next part of the body, and this affects the next part of the body. That was an astonishing observation.
It was astonishing because it seemed too simple. Nobody else thought single genes were powerful enough to control something as complex as the structure of the body. Skeptics argued that Lewis's idea was guesswork. Of course, he had never seen the genes, because the techniques to do so didn't exist.
From the 1920s to the 1970s, it was not possible to physically isolate any specific gene. That opportunity first became available, fortunately for me, at the time that I was a student. And so many of us thought, wow, we can finally dig in there and identify these really mysterious genes.
Levine enlisted his friend and fellow scientist, Bill McGinnis. The first gene they went after had an unusual name, antennapedia, which means antenna leg. The gene was thought to control the growth of legs. When the gene misfired,